So we're going to talk about transport. So to start off with, we're going to talk about simple diffusion. So simple diffusion would be, for example, if you have a cell here. Say, for example, this was just a simple animal cell, and we had oxygen molecules outside the cell, like so. So currently, outside the cell is where we have our high concentration of oxygen, and inside the cell is currently where we have a low concentration of oxygen. So these oxygen molecules would diffuse through the plasma membrane, which they're able to pass through until an equal concentration is established. So we'd end up with an equal concentration. So this is not going to require any energy and doesn't require any proteins in the membrane. So you could have, say, for example, carbon dioxide or oxygen, or you could have, for example, lipid-based hormones which are able to cross the fatty acid component of the phospholipid bilayer. So then what happens is we can have a situation where we might have a high concentration outside the cell of some particular particle, molecule, or substance, and a low concentration inside the cell. But let's say, for example, in this case, the high concentration outside the cell was something like ions, and ions are polar and so therefore cannot cross the plasma membrane. And so then what happens to happen in this scenario is that you're going to need either a protein channel or a protein carrier, depending on what is being crossed. So in this case, you're going to have what is called facilitated diffusion, which is going to be diffusion of substances from an area of high concentration to low concentration with the use of a carrier protein or a channel protein. So a channel protein would be how ions are transported across, and this would be a water-filled channel protein, and a carrier protein would be an example of how, say, for example, glucose, which is too big to go through the plasma membrane, would be transported. So then next what we have is osmosis. So osmosis is going to be the movement of water. So osmosis is the movement of water, and I'm going to choose a root hair cell to demonstrate this. So if a root hair cell has some ions in it, like so, which we can call solutes, then we can say that in the root hair cell there's a high concentration of solutes inside the cell and a low solute concentration outside the cell. So what's going to happen in this scenario is that we are going to have water, which is currently outside the cell and also a bit inside the cell, being drawn into the area of high solute concentration. So that the water is moving to the more concentrated solution inside the cell. And we can say this in a couple of different ways. We could say that osmosis is the movement of water to a high solute concentration, or we could say that the water outside the cell at the moment has a high water potential. So what ends up happening is the water can either move from a high water potential to a low water potential, or we could say that the water moves to an area of high solute concentration. Or we could phrase that the water moves from a relative high water concentration, which is you can see outside the cell, to a relative low water concentration that you can see inside the cell. So osmosis can be defined in a few different ways. It's just important we're aware of those different ways. So we can say osmosis is the movement of water from a high water potential to a low water potential, or you could say to an area of high solute concentration, or you could say from relative high H2O concentration to relative low H2O concentration. And these three definitions are all going to be related to crossing our partially permeable membrane, which is our plasma membrane. Okay, so how does osmosis in, say, for example, a root hair cell actually occur? Well, in order for osmosis to occur, we need to have established this higher solute concentration inside the cell than outside of the cell. And that wouldn't have been possible with just simply diffusion, because diffusion would only leave you with an equal concentration outside and inside. So in order to establish a concentration gradient, say for example with the absorption of ions into the root hair cell from the soil, you're going to need to take solutes against their concentration gradient. So say for example, if these solutes here, these ions needed to enter the cell, they're going to have to go from a low concentration to a high concentration. So they're going to have to go against their concentration gradient. Unlike diffusion, which is down the concentration gradient from low to high. So in order for this to happen, we carry out a process of active transport. And active transport is going to require the use of carrier proteins embedded in the plasma membrane. And these carrier proteins are also going to have sites on them which bind ATP to be converted to ADP for the conformational change of the carrier protein to then allow the glucose to be taken into the cell. So active transport is going to require the use of a carrier protein. It's going to need energy, which is going to be released from ATP converting into ADP to cause the conformational change of the carrier protein, allowing the binding of glucose to enter into the cell so that we can bring in substances against their concentration gradient. So if this is our solute here, being drawn in against its concentration gradient into the cell. Okay, then what we might have is we might have a large amount of substances that need to be carried into the cell, and when this occurs, the cell carries out a process called endocytosis. So endocytosis is going to be when the plasma membrane surrounds and engulfs a particular substance, like so, and then ends up budding into an internal vesicle, which can then be transported around the cell to the relevant place. Now, this process of the plasma membrane moving and the formation of the vesicle is going to be moved on the cytoskeleton, and the cytoskeleton, for transport of these on the cytoskeleton, you're going to need the use of a motor protein. And so if a motor protein is going to be used, that's also going to release ATP to ADP, so it's going to require the release of energy for that to occur. Exocytosis is just going to be the opposite. So exocytosis is when you're going to have a vesicle budding to the membrane and then releasing its contents out of the cell. And again, that is also going to require the use of a motor protein, and these methods are for bulk transport. So example with exocytosis would be, say for example, the secretion from cells, and one of the ones that you may be aware of is the secretion of a neurotransmitter at the synapse of a neuron. Okay, so let's see if we can consolidate some of our understanding of this. So if you pause the video and on a piece of paper try and make yourself a table which looks similar to this, and let's see if we can fill it out. So this is going to be simple diffusion. If we have facilitated diffusion, so I'm just going to write F and have more space. Osmosis, we're going to have active transport, AT, endocytosis, and exocytosis. So we're going to have the movement of molecules from high to lower concentration, same with this, from high to low concentration, and then this is also going to be from high water potential to low water potential. From active transport, we're going to have from low concentrations to high concentrations, and this is going to be from outside to inside, and this is the other way around, inside to outside. So protein in the membrane, none required. Protein in the membrane, channel and carrier proteins. Osmosis, we're going to have aquaporin, which can increase the uptake of water. Special water channel proteins. Active transport is going to require carrier proteins, which are also going to act as enzymes because they transfer ATP to ADP, causing a conformational change of the protein, allowing the iron to enter the cell. Endocytosis, you will require a protein in the membrane, neither with exocytosis. Energy required, no. Simple diffusion, no. Facilitated diffusion, no. For osmosis, all of those are down the concentration gradient. Active transport, yes, that is up the concentration gradient or against the concentration gradient. Endocytosis, yes, for the motor proteins to move the cytoskeleton, to move the vesicle along the cytoskeleton. So examples, we could have CO2 or oxygen or lipid-based hormones. Facilitated diffusion, you could have glucose or insulin or ions, so things that are either too big, polar or lipid insoluble. Osmosis,